Today, we will be covering the topic, the gram-negative bacteria. So generally, these prokaryotic cells, they show a wide range of forms and share some common fundamental traits, and that includes, the first one is the thick complex outer envelope, and their function is to protect the cell from environmental stress and predators. The second one is the compact genome. So, this prokaryotic genome are compact with relatively little non-coding DNA. And finally, the tightly coordinated cell functions. So, in this, the cell's subcellular parts work together in a highly coordinated mechanism which enables high reproduction rate. So, now, as we all know from the previous class that bacteria are classified as gram-positive and gram-negative based on their cell wall properties. So, these bacteria that do not retain the crystal violet stain or the primary stain during gram staining is the gram-negative bacteria, and hence they appear pink in color because of the counter stain safranin, and this is all because of the nature, that is the physical nature of the cell wall. Here, bacterial cell wall is important because it maintains the shape of the cell, and also at the same time it protects the cell from swelling and rupturing. And the gram-negative cell, that is the gram-negative bacteria, may be either spherical, which we also call it as coccus, and another one is the rod shape, which also known as bacillus, coma shaped, also known as curved rods, spiral, also known as spirochetes, or filamentous. So here, in the case of the spiral bacteria do not form association with other cells, and they swim singly through their environments. Their complex structure, that means the spiral bacteria's complex structure within their membrane, allows them to spin their bodies, which propel them along. Further, some rod-shaped and spherical-shaped bacteria forms colonies adhering end to end after they have divided, and they form the colonies or chains. Finally, some filamentous bacteria, they are capable of gliding motion, often combined with rotation around a longitudinal axis. So now we'll come to the biochemical composition of bacteria. In addition to this, all cells, they also share some chemical components, which include, the first one is water. So water, we all know that this is the fundamental solvent of life. The second one is essential ions such as potassium, magnesium and chloride ions. Next is small organic molecules such as lipids and sugars that are incorporated into cell structures, and they provide nutrition and catabolism. Next is macromolecules. Under this, nucleic acids and proteins, which contains information, catalyze reaction and mediate transport. So next we'll come to the gram-negative cell wall. In gram-negative bacteria, the cell wall is composed of a single layer of peptidoglycan, and this is surrounded by a membranous structure, which we termed it as the outer membrane. In this gram-negative bacterial cell, the wall is thinner, that is 10 nanometer thick, and less compact when you compare that with the gram-positive bacteria. But it remains strong tough and elastic to give them shape and protect against extreme environmental conditions. And this peptidoglycan layer, which is present in a very thin layer, is non-covalently anchored to lipoproteins through their hydrophobic head. That was about the cell wall. Now we'll come to the membranes of gram-negative bacteria. So the gram-negative bacterial cell is composed of a thin layer of peptidoglycan, which is found to be in a single sheet. Further, this peptidoglycan layer is covered by an outer membrane which confers defensive abilities and toxigenic properties on many pathogens. Say for example, the Salmonella species and enterohemorrhagic E. coli. So, the gram-negative bacterium is composed of two membranes, that is, the inner membrane and the another one is the outer membrane. So, this inner membrane is called the plasma membrane, and the plasma membrane is a trilaminar structure which is composed of phospholipid bilayer and that surrounds the bacterial protoplasm. So, coming to the outer membrane, the outer membrane also forms a trilaminar structure and consists of proteins such as porins, receptors and asymmetric distribution of lipids. The outer leaflet is composed of lipopolysaccharide and the inner leaflet consists of phospholipid and lipoproteins. The lipopolysaccharides are the integral component of the outer membrane of gram-negative bacteria. And these lipopolysaccharides, they participate in various membrane functions that are essential for bacterial growth and survival. And further, the main function of the outer membrane is to serve as a protective barrier which prevents or it slows the entry of the bile salts, antibiotics and other toxic substances that might kill or injure the bacteria. Also, the outer membrane is more permeable than as compared to the plasma membrane, which permits the passage of small molecules like glucose and other monosaccharides. So, this membrane permeability is mainly due to the presence of porin proteins. Lipopolysaccharides also represents the endotoxins of gram-negative bacteria and they also play a very important role in pathogenesis. So, an endotoxin is a cell component that is harmless as long as the pathogen remain intact. But when released by a live cells, endotoxin overstimulates host defenses, inducing potentially lethal endotoxic shock. Thus, antibiotic treatment of a lipopolysaccharide containing pathogen can kill the cells, but also can lead to the death of the patient. Now we'll see the lipopolysaccharides of the gram-negative bacteria. This lipopolysaccharide is also termed as LPS. So, LPS are also classified into three subunits that includes the first one is the lipid A, second is the core polysaccharide, and the third one is the O side chain or O antigen. So, see here the typical diagram of the lipopolysaccharide. So, this whole structure is the lipid A. Then above that, we have this core polysaccharide. This is the core polysaccharide. And finally, on top of it, it is the O antigen. So, here first we'll come to the lipid A portion. This particular part is the sugar. And this portion is the lipid. When we come to the composition of lipid A, this lipid A is composed of phosphorylated and acetyl glucosamine along with the fatty acids. So when we come to these fatty acids, all the fatty acids that are present in the gram-negative membrane, they are all saturated. That is the saturated fatty acids. And some of these saturated fatty acid, they bound to this phosphorylated and acetyl glucosamine. And some of this fatty acid, they are esterified to 3-hydroxy fatty acid. Here, when we come to the composition, it is composed of phosphorylated and acetyl glucosamine and the fatty acids. And biological function of this lipid A is that these lipid A, they plays a very, very important role in pathogenesis. That is, the toxicity of the lipopolysaccharide generally is conferred by this lipid A portion. But as we all know that this portion of lipid A is intact, that is embedded inside the cell. So it is not exposed as compared to the core polysaccharide and the O antigen. So in order to confirm it, in order to show its toxicity, in order to generate its toxicity, that is the toxicity of the lipid A, this lipid A has to be exposed. So one very important point to be noted is that when the bacteria is alive, when the bacteria is living, this lipid A portion is not exposed. So their toxicity cannot be generated. So in order to generate its toxicity, you need to lyse the bacteria. You need to kill the bacteria. Then only this lipid A portion will be exposed and their toxicity will be generated. So next is the core polysaccharide. This core polysaccharide also, it is composed of sugars. Sometimes some of the proteins and amino acid may also be present, but it depends on which type of gram-negative bacteria it is. Then a very unusual sugar which is present in the core polysaccharide of the gram-negative bacteria is the heptose sugar. So next, coming to the O antigen or the O side chain. O antigen or O side chain is also composed of the sugar molecule. So when we compare this core polysaccharide with the O antigen structurally, this O antigen is far much more longer than the core polysaccharide. And coming to the biological function of this O antigen, it serves as an antigenic determinant. So here, when I say this antigenic determinant, it means it is also immunologically, it is termed as an epitope. So antigenic determinant or the epitope are nothing but these are the portion on the antigen which the immune system recognize. So this immune system may be your antibodies, or it may be your B cell, or it may be your T cells. So next we'll come to the proteins of the outer membrane. So outer membranes also contain a unique protein that are usually absent in the inner membrane. They contain a class of transporters known as porins. So these porins, it permits the entry of nutrients like sugar and peptides. And they also have a distinctive cylinder of beta sheets, which we also termed it as beta barrel. And these gram-negative cells, they also express different porins under different environmental conditions. Cells express a large pore size porins in the dilute environment, thereby maximizing the uptake of nutrients. In contrast, cells express porins of smaller pore size in the rich environment and thereby selecting only smaller nutrients and avoiding the uptakes of toxins. Hence, the porin regulation system of the gram-negative bacteria enables them to grow in an intestinal region that contains the bile salt. So next, we'll see the periplasm of gram-negative bacterial cell. The outer membrane of the gram-negative bacteria is permeable to most ions and many small organic molecules, but it prevents the passage of proteins and other macromolecules. And thus, the periplasm is a membrane-bound compartment of gram-negative cells which lies between the outer and the inner membrane. Also, this periplasm, they contains enzymes and nutrients transporters which are not found within the cytoplasm, such as periplasmic binding proteins for sugars, amino acid or other nutrients. So these periplasmic proteins are subjected to fluctuation in pH and salt concentration as the outer membrane is porous to ions. So next we'll come to the capsule of gram-negative bacteria. The cell wall of gram-negative bacteria contains less peptidoglycan and close by the outer membrane, that we all know. Its outer membrane confers defensive abilities and the toxigenic properties on many pathogens. The outer membrane is composed mainly of the phospholipid and the lipopolysaccharides, which possess the ability to generate a thick capsule surrounding the cell. The capsule polysaccharides forms a slippery mucus layer that inhibits phagocytosis by macrophages. So now we'll come to the gram-negative bacteria that are included in the Burgess Manual Volume 1. So the first one comes the spirochetes. So bacteria under this are flexible and are helical in structure. They possess periplasmic flagella which is meant for locomotion. So most of them are saprophytes or sometimes it may also be parasitic in nature. The second one is aerobic, microaerophilic, motile, helical or vivroid gram-negative bacteria. And the cells under this or the bacteria under this are rigid and move by the help of polar flagella, that is flagella that present towards the ends of the bacteria. And they undergo oxidative metabolism and they are saprophytic or parasitic in nature. Next coming is the third one, that is non-motile or rarely motile gram-negative curved bacteria. So under this, the bacteria are rigid, curved and helical in shape. They lack flagella and are saprophytic in nature. In the fourth point are the aerobic gram-negative rods and cocci. So the cells under this are rigid and straight or slightly curved rods and cocci. They also undergo oxidative metabolism and are saprophytic or parasitic. Next is the facultative anaerobic gram-negative rods. So the bacteria under this are rigid and straight or curved rods. They undergo both oxidative and fermentative type of metabolism and are usually saprophytic or parasitic in nature. Next is the aerobic gram-negative straight, curved and helical rods. So the cells of the bacteria under this group are rigid. They obtain their energy by fermentation or by anaerobic respiration, that is in the absence of oxygen, that does not utilize, they don't utilize sulfur compounds as the electron acceptor. They are mostly parasitic in nature. The seventh one is the dissimilatory sulfate or sulfur reducing bacteria. Bacteria under this are rigid and are mostly anaerobic. They use sulfur compounds as electron acceptors. And most of the bacteria under this are parasites or saprophytes. Next is the anaerobic gram-negative cocci. So bacteria under this, they are rigid and non-motile, and they undergo fermentative metabolism and are usually parasitic in nature. Next is the rickettsias and the chlamydias. Bacteria under this group are rigid and are tiny cells. They are intracellular, that is inside the cell, intracellular parasites of humans, animals and arthropods. The 10th one is the mycoplasmas. The cells of this group are soft and plastic in nature, and most of them are non-motile and lack cell wall. So the bacteria under this are mostly parasitic or saprophytic in nature. Finally is the endosymbionts. Bacteria under this are obligate parasites, that is the strict parasites of protozoa, arthropods or other hosts. So from the above discussion, we can conclude that the gram-negative bacteria are those that do not retain the primary stain, that is the crystal violet, during gram staining. They appear pink in color because of the counterstain safranin, and this is all due to the physical nature of the bacterial cell wall, that is a gram-negative bacterial cell wall. And as we all know that the cell wall of the gram-negative bacteria, they contain less amount of peptidoglycans and large amount of lipopolysaccharides. These lipopolysaccharides is one of the integral component of the outer membrane, and they participate in the various membrane functions that are essential for bacterial growth and survival. Also, these lipopolysaccharides, they play a very important role in pathogenesis, and these biological activity are conferred by the lipid component of lipopolysaccharide, which we also termed it as lipid A.